Good morning, students. Welcome to B.Sc-I second semester Zoology online class. So today I'm going to discuss on the human embryonic development. Human embryonic development. So throughout this chapter, we will express embryonic and fetal ages in terms of weeks from fertilization, commonly called conception. So usually fertilization in human beings is referred as conception. Here the embryonic ages are called by different terms. The period of time required for full development of a fetus in uterus is referred to as gestation. So full term that is required for the completion of development of the embryo inside the uterus is called gestation. Gestation or pregnancy period. So this is the gestation period is nothing but total period required to complete the embryonic development inside the uterus. So gestation, gester meaning is to carry or to bear. So it can be subdivided into distinct gestational periods. See, the gestation is again divided into different gestational distinct periods. So the first 2 weeks of parental development, prenatal development, the first 2 weeks of prenatal development are referred to as the pre-embryonic stage. 2 weeks, up to 2 weeks after the fertilization is referred as the pre-embryonic stage. So from day 1 of fertilization up to the second week of fertilization is referred as pre-embryonic stage. Up to 2 weeks, it would be referred as, the embryo would be referred as pre-embryonic stage. After 2 weeks, the developing human is referred to as an embryo during weeks 3 to 8. From 3rd to 8th week, the embryonic stage is known as embryo. It is referred as embryo. Before that, pre-embryonic stage, embryonic stage, between 3rd to 8th week of the pregnancy. During that period, the growth of the embryo known as, it is in embryo stage. Then third stage is from 8th week onwards up to the birth of the child. Parturition, that would be referred as fetus. From the 9th week of gestation until birth. From 9th week of gestation up to the birth of the child. So thus the gestation period, total gestation period or pregnancy period has been divided into 3 stages: pre-embryonic stage, embryonic stage and fetal stage. Pre-embryonic up to second week, then from 3rd to 8th week embryonic stage, from 9th up to the birth of the young one, the child, until the birth of the child would be referred as fetus. In this section, we will cover the pre-embryonic and embryonic stages of development, which are characterized by cell division, migration and differentiation. Right? So here we are going to study 2 stages, embryo of 2 stages, pre-embryonic stage and embryo stage. So here they show, the embryo shows different embryonic developmental properties: cell division, migration of the cells, differentiation of cells. By the end of the embryonic period, all of the organ systems are structured in a rudimentary form. By the end of embryonic period ends by 8th week of the pregnancy. By that time, all the important organ systems are developed, but they are in a rudimentary condition. although the organs themselves are either non-functional or only semi-functional. So next, pre-implantation embryonic development. Pre-implantation embryonic development or embryonic development. Before implantation. Okay. So following fertilization, the zygote and its associated membranes together referred to as conceptus. After fertilization, the zygote and its associated membranes together referred to as conceptus. Okay. So continue to be projected toward the uterus by peristalsis and beating cilia. During its journey to the uterus, the zygote undergoes 5 or 6 rapid mitotic cell divisions. After fertilization, right, the zygote would be moving towards the uterus. Look here. So here fertilization occurs. Fertilization. So this is the ovum. Fertilization. This is ovum and these are sperms. Sperms enter into the ovum. Only one sperm enters. Only one sperm enters. This is called as monogamy. Monospermy. Sorry, monospermy. Only one sperm can fertilize the ovum. So this is fertilized oocyte. After fertilization, it moves towards the uterus. While it is moving towards the uterus, the zygote undergoes repeated cleavage, mitotic division, and this is not moving on its own because ovum is not having its own locomotory organelle. Therefore, it is pushed towards the uterus by the beating up of cilia present in the wall of the fallopian tube. So now the zygote undergoes first cell division. So 2 cell stage, it is 2 cell stage, then 4 cell stage, then 8 cell stage, morula. When it comes here, it would be having 16 cells. Then when it reaches the cavity of the uterus, this undergoes again cell division. It may have around 70 to 100 cells. When it comes to the uterus, cavity of the uterus, it would be having around 100 cells, and this is called as blastocyst. What is this? Blastocyst. Blastocyst would be like this. Okay. So this ball-like embryo having cavity at the center. This cavity is called blastocele, and this peripheral layer of cells is called as trophoblast. And inside the cavity, some cells grouped and they are attached to one part of the at one side of the trophoblast, and this is called inner cell mass, and this is cavity. This cavity is called blastocele. So this is nothing but blastula stage of the human embryo, but it is referred as blastocyst. Instead of blastula, it is being referred as blastocyst. Now this blastocyst, what it does, it attaches to the wall of the uterus, and that would be referred as implantation. Okay. So in this section, we will cover the pre-embryonic and embryonic stage of development, which are characterized by cell division. Okay, this is over, and here we were here. So zygote undergoes 5 or 6 rapid mitotic cell divisions. Although each cleavage results in more cells, it doesn't increase the total volume of the conceptus. total volume of the conceptus. What is conceptus? Conceptus is nothing but zygote along with its membrane. So externally it is covered by zona, the what do you call fertilization membrane. Zona pellucida is there. Zona pellucida plus zygote, both together known as conceptus. Okay. So the conceptus volume is not increasing during a cleavage. So although each cleavage results in more cells, it doesn't increase the total volume of the conceptus. So each daughter cell produced by cleavage is called a blastomere. Each cell which is produced by the cleavage is known as blastomere. So blastose, germ, in the sense of seed or sprout. So approximately 3 days after fertilization, a 16 cell conceptus reaches the uterus. I have already shown you. So here, so when the embryo would be having 16 cells, it has reached the cavity of the uterus. Later on, again it undergoes further cleavage and its cells number is increasing. So approximately 3 days after fertilization, 16 cell conceptus reaches the uterus. The cells that had been loosely grouped are now compacted and look more like a solid mass. It looks like a solid mass. The name given to this structure is called morula. Morula is a ball-like cellular embryo which is not having cavity at the center. It's a solid ball of cells. Morula. It is little mulberry. Mulberry fruits. Mulberry fruit like embryo it is. Therefore, it is called as, this stage is known as morula stage. Once inside the uterus, the conceptus floats freely. Once inside the uterus, the conceptus floats freely for several more days. It continues to divide, creating a ball of approximately 100 cells. A ball of approximately 100 cells and consuming nutritive endometrial secretions. So the embryo is consuming nutritive substance. So nutritive substance secreted in this cavity by the endometrial lining. This is called endometrial lining. Endometrium it is. So epithelium of the endometrium secretes secretion. That secretion is rich in nutrients. They are entering into the blastocyst. Okay. So consuming nutritive endometrial secretions called uterine milk. This is called as uterine milk. Okay. So uterine milk is nothing but secretion of the uterine lining, endometrium. So endometrium secretes some secretion that would be referred as uterine milk, and it is rich in nutrients. That is going into the blastocyst while the uterine lining thickens. So next, the ball of now tightly bound cells starts to secrete fluid and organize themselves around a fluid-filled cavity, the blastocele. At this developmental stage, the conceptus is referred to as blastocyst. Okay. So cells are arranged in a layer around the central cavity, and that is called trophoblast, and the central cavity is called blastocele, and this stage is called as blastocyst, not blastula. Blastocyst. In case of frog, it would be referred as blastula, but here in human beings, the blastula stage is called blastocyst. Within this structure, a group of cells forms into an inner cell mass. Yes. So into the cavity, there is a mass of cells called inner cell mass, which is fated to become the embryo. So that mass only going to be developed into embryo. The cells that form the outer shell are called trophoblast. So these are called trophoblast. So these cells are called trophoblast cells. And these cells together form inner cell mass, and this mass is going to be developed into actual embryo, not this trophoblast. Trophoblast is used for development of placenta. Okay. So these cells will develop into the chorionic sac and the fetal portion of the placenta, trophoblast cells. So they are going to be developed into chorion, allantois, yolk sac membrane. These are all called as extra embryonic membranes. They are formed by the outer cells present in the trophoblast cells, chorionic sac and the fetal portion of placenta. See, the placenta is the organic connection between the embryo and the mother. So this placenta is made by the investment of tissues from mother side as well as fetal side. So fetal tissues, these are fetal tissues invested for the formation of placenta, used for the formation of placenta. So the organ of nutrient waste and gas exchange between mother and the developing offspring. So this placenta helps in the exchange of substances between maternal and fetal blood circulation. So nutrients are supplied from mother to fetus. Wastes are removed from fetus and they enter into the maternal circulation. Gas exchange is also being done through this placenta. The inner mass of embryonic cells is totipotent during this stage. So inner mass of cells, embryonic cells, are totipotent. So these cells are totipotent in nature. Totipotent means these cells can be developed into any type of tissue, that potential they have. Therefore they are called totipotent cells. Okay, so there are different types of potency having cells: pluripotent, totipotent. Okay, so these are totipotent cells at this stage. After this stage, the cells may lose this totipotency. Okay. So inner mass of embryonic cells is totipotent during this stage, meaning that each cell has the potential to differentiate into any cell type in the human body. Totipotency lasts for only few days before the cell's fates are set as being the precursors to a specific lineage of cells. So before the cells become the precursors of any organs, these cells would be totipotent in nature. Okay, so this is 2 cell stage, 4 cell stage, 8 cell stage, morula stage, so blastocyst. Okay, so this is having around 100 cells. So it is still externally covered by zona pellucida. Okay, when it becomes blastocyst, it floats in the milk, uterine milk, for a few days. Later on, the zona pellucida is ruptured. This blastocyst is released into the cavity of the uterus. So as long as it is covered by zona pellucida. It is unable to adhere to the wall of the uterus. When the zona pellucida is removed, the blastocyst can adhere to the wall of the uterus. That process of attachment of blastocyst to the wall of uterus is called implantation. Implantation. Okay. Now pre-embryonic cleavages. So far, yes, pre-embryonic cleavages. So these are pre-embryonic cleavages. Pre-embryonic cleavages make use of the abundant cytoplasm of the conceptus as a cell rapidly divides without changing the total volume. As the blastocyst forms, the trophoblast excretes enzymes that begin to degrade the zona pellucida in a process called hatching. Okay, zona pellucida, I told you. Zona pellucida is a layer present around the blastocyst. This is ruptured. This process is called as hatching. The conceptus breaks free of the zona pellucida in preparation for implantation. Now it is going to be prepared for implantation. Implantation means, I told you, attachment of blastocyst to the wall of uterus. So implantation. So cleavage and implantation occur during the pre-embryonic stage. Okay. Now the uterine cavity has blastocyst. What it has? Blastocyst. Now blastocyst has to be attached to wall of the uterus for getting requirements such as nutrients, then respiratory gas like oxygen. Okay, whatever substances required by the embryo, they are being supplied from the maternal circulation to the fetal circulation. So for that sake, it has to be attached to the wall of uterus, and that is called implantation. Now let us discuss what is implantation in detail. At the end of the first week, remember, at the end of the first week of fertilization, now 1 week is over. So first 2 weeks embryonic stage would be referred as what? Pre-embryonic stage. What is this? Pre-embryonic stage. Now 1 week is over. 1 week is over after the fertilization. Now it is in a cavity of the uterus. The blastocyst comes in contact with the uterine wall and adheres to it, embedding itself in the uterine lining via the trophoblast cells. Thus begins the process of implantation, which signals the end of pre-embryonic stage of development. This process, the process of implantation takes around 1 week time. So first week the embryo has been reached the uterine cavity. Later on it gets attached to the wall of the uterus, and that attachment continues for another 1 week. And this process of implantation completion indicates the completion of pre-embryonic stage. So look here, this is blastocyst. Now blastocyst is attached to uterine wall. This is what? Uterine wall. Now it has attached. Okay, so where it is attached? Where actually inner mass of cell is present, attached to the trophoblast. That trophoblast comes in contact with the uterus. So this is uterine cavity. This part is uterine cavity. This is endometrium. What is this? Endometrium. This is what? Endometrium epithelium. And this epithelium is ruptured. epithelium of the endometrium is ruptured by the secretion of some secretions, okay, by the trophoblast cells. So the blastocyst digests uterine mucosa when it initially implants into the endometrium. So blastocyst digests the endometrial lining here. ಸೊ ಇಲ್ಲಿ ಏನಾಗ್ತದೆ, ಇಲ್ಲಿ ಕಟ್ ಆಗ್ತದೆ, ಇದು ಎಪಿಥೀಲಿಯಂ ಕಟ್ ಆಗಿಬಿಡ್ತದೆ. ಎಲ್ಲಿ ಇದು ಅಟ್ಯಾಚ್ ಆಗಿರ್ತದಲ್ಲ, ಅಂಟ್ಕೊಂಡಿರ್ತದಲ್ಲ, ಅಲ್ಲಿ ಇಲ್ಲಿರುವಂಥ ಸೆಲ್ಸ್ ಎಲ್ಲ ಡೈಜೆಸ್ಟ್ ಆಗಿಬಿಡ್ತಾವೆ. ಇದು ಗ್ರ್ಯಾಜುವಲಿ ಒಳಗೆ ಹೋಗ್ತದೆ. ಇಟ್ ಸಿಂಕ್ಸ್ ಇನ್ ಟು ದ ಎಂಡೋಮೆಟ್ರಿಯಮ್. It gradually sinks into the endometrium. So this is having endometrial epithelium. I mean the tissues. ಈ ಟಿಶ್ಯೂ ಒಳಗೆಲ್ಲ ಇದು ಒಳಗೆ ಹೋಗ್ತದೆ, ಮುಳುಗ್ತದೆ. ಇದು ಇಲ್ಲಿ ನೋಡಿ, ಇಟ್ ಸಿಂಕ್ಸ್, ಒಳಗೆ ಮುಳುಗ್ತದೆ. ಮುಳುದಾಗ ಏನಾಗ್ತದೆ ಗೊತ್ತಾನೆ, ಈ ಎಪಿಥೀಲಿಯಮ್ ಇದ್ರ ಮೇಲೆ ಕವರ್ ಆಗ್ತದೆ, ಕಾಣ್ತದೆ. ಅದರತ್ತೇನು, ದ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಈಸ್ ಕಂಪ್ಲೀಟ್ಲಿ ಸಂಕ್ ಇನ್ ಟು ದ ಎಂಡೋಮೆಟ್ರಿಯಮ್. ಇದು ಪೂರ್ತಿ ಮುಳುಗಿಬಿಟ್ಟದ. ಇದ್ರಾಗ ಮುಳುದಾಗ ಏನಾಗ್ತು, ಇದ್ರ ಮೇಲ್ಗಡೆ endometrial lining is developed, endometrial epithelium. Eventually the endometrium grows over and surrounds the embryo, fully securing it to the uterine lining. ಇದು ಪೂರ್ತಿ ಸೆಕ್ಯೂರ್ಡ್ ಆತಿಗೆ. ಇದು ಪ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಪೂರ್ತಿ ಒಳಗಡೆ ಹೋಯಿತು. ಒಳಗಡೆ ಈ ಕ್ಯಾವಿಟಿ ಒಳಗಿಲ್ಲ. ಈ ಕ್ಯಾವಿಟಿ ಒಳಗೆ ಏನದ, ಯೂಟ್ರೈನ್ ಮಿಲ್ಕ್ ಅದ, ಗೊತ್ತಾಯ್ತು. ಇದು ಒಳಗಡೆ ಪೂರ್ತಿ ಹೋತು. ಇದ್ರ ಮೇಲ್ಗಡೆ ಎಪಿಥೀಲಿಯಂ ಬೆಳೀತು, ತಿಳಿತು. ಇದು ಸೆಕ್ಯೂರ್ ಆತು, ಪೂರ್ತಿ ಸೆಕ್ಯೂರ್ ಆತು. ಇದಕ್ಕೆ ಇದು ಅಂಟ್ಕೊಂಡ್ಬಿಡ್ತು, ಪೂರ್ತಿಯಾಗಿ ಅಂಟ್ಕೊಂಡ್ಬಿಡ್ತು. ಅಂಟ್ಕೋತಂದ್ರೇನು, ಇದ್ರಾಗಿರುವಂಥ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಎಲ್ಲ ಇದಕ್ಕೆ ಸಪ್ಲೈ ಆಗ್ತಾವೆ, ಇದಕ್ಕೆ ಡೈರೆಕ್ಟಾಗಿ ಸಪ್ಲೈ ಆಗ್ತಾವೆ. ಇನ್ನೇನಾಯಿತು, ಇವುಗಳ ಮಧ್ಯೆ ಒಂದು ಇಂಪ್ಲಾಂಟೇಷನ್ ಮುಗೀತು. ಈಗ ಇಂಪ್ಲಾಂಟೇಷನ್ ಆದ ನಂತರ ಪ್ಲಾಸೆಂಟಾ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತದೆ. ಪ್ಲಾಸೆಂಟಾ ಅಂತೇಳಿ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ನೋಡಿರಿ, ಇಲ್ಲೇ ಇದೆ ನೋಡಿರಿ, ಇಲ್ಲಿ ಕಾಣ್ತದೆ. ಇದು ಎಂಬ್ರಿಯೋ. ಈ ಎಂಬ್ರಿಯೋದಕ್ಕೂ ಮತ್ತು ಈ ಎಂಡೋಮೆಟ್ರಿಯಮ್ಗೂ ಒಳಗಡೆ ಯೂಟ್ರಸ್ಗೂ ಕನೆಕ್ಷನ್ ಐತಿ. ಇದಕ್ಕೆ ಅಂಬಲಿಕಲ್ ಕಾರ್ಡ್ ಅಂತಾರೆ, ಅಂಬಲಿಕಲ್ ಕಾರ್ಡ್. ಇಲ್ಲೇನದ ನೋಡಿರಿ, ಈ ಭಾಗ, ಇದು ಪ್ಲಾಸೆಂಟಾ. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಪ್ಲಾಸೆಂಟಾ. ತಿಳಿತು. ಇದು ಪೂರ್ತಿ ಎಂಟೈರ್ ಎಂಬ್ರಿಯೋ ಎಲ್ಲದ, ಇಟ್ ಈಸ್ ಕವರ್ಡ್ ಬೈ ಎಂಡೋಮೆಟ್ರಿಯಮ್. ಕ್ಯಾನ್ ಸಿ ಹಿಯರ್, ಇದು ಪೂರ್ತಿ ಕವರ್ ಆಗಿದೆ. ಹೌದಿಲ್ಲ, ಇದು ಪೂರ್ತಿ ಎಂಡೋಮೆಟ್ರಿಯಮ್ದಾಗ ಆಗ್ತೈತಿ. ಇಲ್ಲ, ಹೊರಗಡೆ ಇರೋ ಕ್ಯಾವಿಟಿ ಇದು ಕ್ಯಾವಿಟಿ. ಈ ಕ್ಯಾವಿಟಿ ಕಡಿಮೆ ಆಗ್ತದಿಲ್ಲ, ಇದು ಬೆಳೆದಂಗೆ ಇದು ಕ್ಯಾವಿಟಿ ಕಡಿಮೆ ಆಗ್ತದೆ. ಆ್ಯಸ್ ಇಟ್ ಗ್ರೋಸ್, ಆ್ಯಸ್ ದ ಎಂಬ್ರಿಯೋ ಗ್ರೋಸ್, cavity size diminishes. Okay, so the entire embryo is covered by endometrium. Okay, so thus begins the process of implantation, which signals the end of the pre-embryonic stage of development. Implantation can be accompanied by minor bleeding. So minor bleeding may occur during implantation. The blastocyst typically implants in the fundus of the uterus, in the upper part of the uterus, or on the posterior wall. However, if the endometrium is not fully developed and ready to receive the blastocyst will detach and find a better spot. So a significant percentage, 50 to 75% of blastocyst fails to implant. When this occurs, the blastocyst is shed with the endometrium during menses. Okay, if it is not implanted, it would be shed during menses. The high rate of implantation failure is one reason why pregnancy typically requires several ovulation cycles to achieve. So implantation may fail, but sometimes it happens, implantation. So usually during the normal, developmental stage, the implantation occurs. Then after implantation, of course, if implantation doesn't occur, the embryo is, embryo means the blastocyst is shed outside. It is going outside during menses. If the the womb is not fertilized, if it is not fertilized, it will go out. After, of course, even sometimes after fertilization also, it may not adhere to the wall of the uterus. So if it is not able to adhere, it may go out of the uterus through menses. So these are the different stages that occur during the embryonic and pre-embryonic and embryonic stage. Next. So when implantation succeeds and the blastocyst adheres to the endometrium, the superficial cells of the trophoblast fuse with each other, forming the syncytiotrophoblast, a multinucleated body that digests endometrial cells to firmly secure the blastocyst to the uterine wall. So I told you the trophoblast cells, they develop finger-like structures. They enter into the endometrium to form syncytiotrophoblast. In response, the uterine mucosa rebuilds itself and it develops the blastocyst. The trophoblast secretes human chorionic gonadotrophin. Yes, during this time, the trophoblast secretes human chorionic gonadotrophin. Okay, so the trophoblast cells secrete human chorionic gonadotrophin, HCG. So this HCG is very important. So by analyzing HCG in the woman urine, the woman is confirmed that she is pregnant by the presence of HCG in the urine of women. Okay, so a hormone that directs the concept as corpus luteum to survive. So this HCG helps in stimulation of it stimulates corpus luteum to secrete progesterone hormone. Progesterone hormone is necessary for the maintenance of pregnancy. Therefore, the corpus luteum life is extended by the secretion of HCG. So earlier to the secretion of HCG, corpus luteum is stimulated by the LH hormone, luteinizing hormone, secreted by pituitary gland. Okay, now HCG hormone directs the corpus luteum to survive, enlarge, and continue producing progesterone and estrogen to suppress menses. So menses is not occurring during pregnancy till the childbirth. After childbirth also, after 3 to 6 months, there would be no menses as long as the young one suckles the mammary gland. It feeds on mammary glands. It feeds on the milk. The menses may not happen. These functions of HCG are necessary for creating an environment suitable for the developing embryo. So, as a result of this, increased production of HCG accumulates in the maternal bloodstream and is excreted in the urine. It is excreted in the urine. Okay, that is analyzed by during the test of pregnancy. So implantation is complete by the middle of the second week. So middle of the second week, implantation is completed. Just a few days after implantation, the trophoblast has secreted enough HCG for an at-home urine pregnancy test to give a positive result. To give a positive result, HCG is important. If HCG is present in the urine, it is confirmed that the woman is pregnant. So pregnancy is confirmed. Pregnancy positive test is the presence of HCG in urine. So this is how implantation takes place. So during implantation, the trophoblast cells of the blastocyst adhere to the endometrium and digest endometrial cells until it is attached securely. So this caption is because of, this refers to this diagram. These diagrams are explained by this statement. Now, when implantation succeeds and the blastocyst adheres to the endometrium, the superficial cells of the trophoblast fuse with each other, forming. Sorry. So implantation. Okay. So most of the time, an embryo implants within the body of the uterus. Most of the time, an embryo implants within the body of uterus in a location that can support growth and development. However, in 1 to 2% of cases, the embryo implants either outside the uterus, that sometimes it may get implanted in the fallopian tube, that is called as ectopic pregnancy, or in a region of uterus that can create complications for the pregnancy. If the embryo implants in the interior portion of the uterus, the placenta can potentially grow over the opening of the cervix, a condition called placenta previa. That is not necessary. Okay, so with this we have completed the human embryonic development from its after its fertilization. It undergoes cleavage, then it becomes blastocyst. Blastocyst adheres to the wall of the uterus. This is called implantation, and this implantation process is helped by the trophoblast cells, which secrete digestive enzymes which digest the lining of the endometrium. Then it is completely covered by the endometrial lining. Later on, the placenta develops. Okay, implantation is completed. Up to the implantation process, how the human embryo develops, that we have discussed. Let us see the video clipping is there, which gives you an idea of different stages of embryonic growth in human beings. Let us see it.
Journey of life begins with a single cell, but later it is no longer alone. It divides repeatedly to form a mass of many cells together. And in this video, we are going to learn about this beautiful process that helps us grow to what we are from a single cell. Yes, we will be talking about how the embryo develops. Do we have a scientific name for this process of embryo development? Absolutely, it's called the process of embryogenesis. In simplest words, embryogenesis is the process that follows fertilization. Where exactly does the fertilization take place in humans? Yes, it takes place in the fallopian tubes of the female reproductive system. However, before reaching the uterus, the embryo undergoes a series of divisions that help in its growth and development. Let us focus on one of the fallopian tubes where the egg cell has got fertilized. Now this fertilized egg cell will divide to form 2 cells followed by 4 and so on. This results in the formation of a ball-like structure. This ball contains many cells inside it. Do you know what this ball of cells is referred to as? When the cell number in it is 16, then it is referred to as the morula. This further divides to form a structure called the blastula. So will it have double the number of cells in it? Blastula is just a stage where the cells arrange themselves such that they form a hollow mass structure. In fact, the cells align themselves to the ends and leave a cavity within, which gets filled with the fluid. Now the next stage of development will be the blastocyst. It is the stage where the embryo will have cells that have got differentiated and will even differentiate further. Now what exactly is this process of differentiation? Let me explain. Have you ever thought that if we all started our lives with a single cell, then where do all these different types of cells in our body come from? Yes, I know it's from the single cell that was present at the beginning. But how is this possible? The answer to this is the differentiation process. As the name suggests, it is the process in which one cell gets differentiated from the other cells in its vicinity. As a result, we get a whole new variety of cells from these differentiated ones after successive divisions. So coming back to the blastocyst stage, it is now a mass containing differentiated cells. As it grows further, the body of the embryo begins to develop. But for its proper growth and development, don't you think the embryo will require nutrition? Of course, yes. And where will it derive the necessary nutrition from? You may say the mother's body. That's correct, yes. But how will that be possible when the blastocyst is still moving inside the reproductive tract? It is not connected to the point from where it can derive nutrition. Hence, the blastocyst needs to be connected to that particular location. This is where the most important stage of implantation comes into the picture. Can we guess what could it be from the name itself? Yes, it is the step where the blastocyst now attaches itself inside the mother's body at one fixed position. It implants itself there. And where will this position be? Of course, inside the uterus. That is the place where the baby grows, right? So the embryo now attaches itself in the uterus. Few of the main reasons for attachment is deriving nutrition, giving away wastes, and exchanging gases. This is served through this barrier that we can see here. This barrier is an organ called the placenta. So can we say that the placenta connects the baby with its mother? Absolutely. Now the embryo is all set to grow into a baby. The stage where the organs of the baby's body starts developing such that they can be identified is called as the fetal stage. And the embryo can now be called a fetus. The fetus grows in the uterus after taking the required amount of time, and when the growth is completed, the child is all set to enter this beautiful world. This is how an embryogenesis takes place in most of the animals. But
so that is the different stages that happen for the growth of the embryo in human beings. Okay, so that is about the human embryonic growth up to implantation. Now few assignments are there. The first assignment is what is zygote. Second one is where fertilization takes place in female reproductive system, and what is implantation. So these are simple questions, so you have to give answer. Okay, so with this we have completed the embryonic growth in human beings. Thank you.